So, is there a way you can tell a virus from an obligate intracellular bacterial pathogen? For example, I don't know, chlamydia. There are probably two things that people use to distinguish them more or less definitively. One, does the organism bring its own ribosomes with it? Right? And two, is there a boundary? Even when it's infected inside another organism, in the case of an obligate intracellular parasite bacteria, there's always this distinction inside between what's the parasite and what's the host. And so the two are physically separated, even in the cytoplasm of the host. Whereas in a virus, the two are intermingled in some way. Yeah.
You define life by having
I think you wouldn't want to do that because then extraterrestrial life is almost impossible by definition. Right? I mean, you could walk around and some radar signals and so forth, but if it doesn't have ribosomes, if it has a different basis for biology, I think you'd still want to consider it a lot.
we say look at something that's about
Depends on how big your imagination is. So you notice in all the trees I've showed you, viruses aren't included. That may change. Basically, though, there are three possible origins for viruses. One, that they're genetic offshoots of their host. That is, they are rogue genes. Genes that originated in the genome of their host or relatives of the host that have become evolutionarily independent and their own organism, so to speak. I think there are clear examples of this. Two, they may be relics of very, very early forms in the evolution of life. I think there is not good evidence for this. And three, they may be highly reduced remnants of what used to be obligate intracellular organisms. And I think there's increasing evidence for examples of this. Yeah?
Has someone produced some sort of phylogenetic tree of viruses? Not including anything else, but just like how viruses are?
Yes. And what you have to do, so the question is, can you make trees of viruses? And the answer is absolutely yes, but you can only do it within classes of a virus. And so, for example, the T-even phage are all related to each other, and you can make trees all you want with those. The T-odd phage, HIV-like retroviruses, copia element. I mean, so there are these families of viruses that are clearly related. But you can't make trees between the classes because there's no evolutionary ancestry between them. There's no evidence that they ever have anything the same. Yeah. So you really do have to divide the viruses into families and treat them independently. Viruses aren't a phylogenetic group. They're a functional group. They're a collection of things that we drop into this thing called viruses, even if they're not related. And this, I mean, when you look at their structure, this is more or less obvious. They're single-strand RNA viruses or double-strand DNA viruses. No evidence that they're related to each other. And that's what this is about. That you really do have to treat different classes of viruses as different kinds of things. So one of the notions is that viruses or some kinds of viruses might be evolutionary offshoots of their hosts. And so one way people study this sort of a thing, where an unusual group of organisms come from, is to create an evolutionary series. So if, for example, you're baffled by the fact that manta rays are the way they are, you might ask yourself, okay, how could this thing evolve? And then you take a mundane fish and you imagine the evolutionary steps that would take you from a mundane fish, maybe a cartilaginous fish like a shark, all in step by step to make a manta ray. Now those are all hypothetical, right? And they're all ancestors. But you can strengthen that argument and show that they're rational by picking a modern organism that fits that type. And so you take a bunch of modern organisms and you put them in a row as a way of modeling the evolutionary process leading to something. And so you might, for example, start with a pelagic shark, like blue shark, and then you've got carpet sharks, things like wobbegongs and so forth, sawfish, skates, electric skates maybe, and then traditional rays, and then something like a mana ray. And you put them all in a row, and it makes sense that you could evolve from one to one in a step-like fashion. Now, the Kauno's ray is not an ancestor of the mana ray. It's a modern organism, but it's a stand-in for an evolutionary ancestor. Can we do the same thing with with bacterial genomes and viruses, or host genomes of any kind and viruses? And the answer is yes. And so bacteria contain generally a large circular chromosome. Many of them, perhaps most of them, also contain secondary chromosomes. We call these plasmids. Right? These are smaller circles. They often don't contain essential genes on them, at least genes essential for some. Many of these plasmids contain genes that direct the transmission of that secondary chromosome from one cell line to another. Sometimes they carry other genes with them, sometimes they don't. We call these fertility factors, or conjugated plasmids. And so F is the stereotype of this in E. coli. E. coli cells are either F plus or F minus. If they contain the F plasmid, they're F plus, and that F plasmid can transmit to a recipient F minus strain and make it F plus. And it's notorious for picking up chunks of the genome as it does this. It does this by integrating into the chromosome and then re-excising incorrectly and biting off a chunk of the chromosome. And this exchange of DNA from one cell line to another is a form of sexual exchange of genetic information. It's a stand-in for sexual exchange in bacteria. And so F is clearly an offshoot of the host. That's part of the genetics of the strain. What if, though, instead of requiring direct contact to transfer that DNA, you develop a protein that coats the DNA and allows the plasmid, a conjugated plasmid, to be secreted from the cell into the environment and then taken up by the recipient strain without the need for direct contact? There are lots of viruses like this. FD, M13, how many of you have heard of these organisms before? M13 is a common lab thing that people use for cloning and so forth. M13 exists as a double-stranded circle in the host cell. It does not kill the cell. It does have a phenotype. The cells grow a little slower. There's some cost to the host of maintaining this DNA in it, but it's not lethal in any way at all. M13 is really a lot like F. It's just that it encodes a coat protein so that it can be transmitted indirectly. All you have to do is add a lytic gene to this, so that if it's an advantage of the host to reproduce very quickly and lyse the cell, then you've got a lytic virus. There are also some cases of viruses and bacteria that clearly emerge from transposons. And so mu is a great example of this. People think that mu gets its name from the Greek letter. It does not. It's short for mutator. And so this is a transposon that's encapsulated, and when it's injected into the host cell, it transposes into the genome. And when it does so, it tends to knock out genes. And the result is mutations, and so it's used as a mutator. When mu is in the host, it measures the state of the cell. If the cell is metabolically healthy, it stays put. It shuts off transposition, and it sits there quietly because the cell is growing, it's reproducing, and the mu gets reproduced passively as a result, and it ends up in all the offspring of that host. If the cell's in trouble, oftentimes we're measuring things like PPGPG, PPGPP produced by the ribosomes that are stalled, or by the SOS response, or this sort of a thing. If the cell is metabolically in trouble, it would be to the advantage of that transposon to take over and to make as much of itself as it can before this host cell buys it. And the way it does this is that it then turns on the transposase, and mu starts to jump all over the chromosome. It just goes completely nuts. It does a replicative transposition, and it does this over and over and over again, and it fills up the genome with its copies of itself. And the late expression genes then are capsid genes that cut these things out, that they actually transpose out of the genome and then are encapsulated, and when the cell lyses, they're released into the environment, and the process can repeat. Yeah?
They come in homologous genes to the capsid genes in the host genome. Did they acquire them from the host genome?
That's a good question that I don't know the answer to. That would depend probably on the class of the virus. Mu's a little unusual that way. Mu's very complicated. It looks like a real virus. Let's see, do I have a... Yeah, okay. So another example of this are retroviruses. Retroviruses are really nothing more than some control genes of capsid stuff and a reverse transcriptase. The retroviruses are reverse transcriptase genes out of control. TY elements in yeast, copia, P elements, HIV, all of these things are clearly derived from the genetics of the host. It's also been proposed that some viruses might be relics of a pre-cellular RNA world. I'll admit that I don't buy this at all. Nevertheless, the folks who study these things often do talk about this. Well, first of all, it requires that you believe in an RNA world. That is, a time in evolutionary history before the invention of DNA and protein, but after the invention of RNA. This makes some sense because RNA can serve as a genome and it can be catalytically active. And so RNA can carry out both roles of genome and activity, whereas protein and DNA can't do those things. And so if these things were invented independently, RNA would have had to be first for the others. Even for those who believe in an RNA world, the complexity of that is a matter of some debate. And so some people will tell you that there may have been an RNA world, but it was taken over. As soon as you had a self-replicating RNA, it would have started coding for oligopeptides that would facilitate that process, and you would end up with an RNA protein world. Others will tell you that there was a very complicated RNA world containing most of the catalytic activities that we think of today, and that those RNAs have been replaced by protein piecemeal. And that the relics of those RNAs are things we call cofactors, which are almost always nucleotides. FAD, FNN, KPP, all those things. Again, I think outside the realm of scientific study at this point. It's pretty speculative. But there are some really interesting cases today. And so here's the rice yellow model virus-associated viroid. This is one of these viruses of viruses. That's not the genome of this thing. That's the whole thing. Right there. There's no capsid, no accessory factors. That's it. The whole thing. Just a little over 200 nucleotides, and it folds up on itself, and that's it. And this thing is a great substrate for the rice yellow model virus replication machinery itself. So this can only infect rice cells that are infected by the rice yellow model virus, and it takes that other virus's genetic replication machinery, which binds to this thing and replicates it out of control. It's kind of a parasite on the original virus's genetic machinery. Pretty cool. Hepatitis delta virus is another one of these.
If we started with RNA,
Yeah.
like let's say that we get something in RNA, what would have been the benefit to having conversations to having that evolution
DNA is a lot more stable. It's much less subject to chemical and physical damage. Let me give you a couple of examples of these. So here's, I think this is the Delta virus, but it's common. So a lot of these things are RNAs that go back and forth between plus and minus strand, right? And so if you've got a positive strand RNA virus, it goes through rolling circle transcription, and you've got a catalytic RNA encoded on that in that sequence. Pretty often, that negative strand has a so-called hammerhead ribozyme on it. Binds magnesium, and it's self-cleaving. That reaction is reversible. And so if you have a long chain of these things, and they self-cleave and then close up again, kinetically, the most likely outcome of that reverse reaction is for the molecule to close up on itself as a circle. It's unlikely that this end is going to find another end somewhere else. It's much more likely to find something nearby, which is right here. And so when you go through a cleavage and reversal, you end up taking these, people call them paper towel transcripts, and you end up with a bunch of circles. This then serves as a substrate for positive strand replication. The replicase does that, and you do the same rolling circle sort of a thing, and on this strand you have a hairpin ribosome. Same sort of thing, self-cleavage, self-ligating. And this process can go on and on and on in the infected cells. You see this a lot in plants, because in plants, cells have these junctions, direct cytoplasmic connections. And so these RNAs can be transmitted from cell to cell without the need to go out and be exposed to the external environment. They're always in cytoplasm. No. So, I mean, this thing is serving as a genome, right? It's got catalytic activity, right? Imagine that an early RNA developed the ability to do this before it was in a cell. That is, it could replicate in an environmental situation and reproduce in this way. That's a pretty tough sell for me. I don't see you finding an environment. I mean, you might imagine that you could code a replicase in here or something like that, or one of these RNAs could have replicase activity. You can do a little bit of that by in vitro selection. You can prove that that's feasible. The biggest issue is where you're going to get your monomers. UTP just doesn't float around in the environment, right? In all cases where people have studied this, the information content of these things is really low. It's absolutely clear that they spontaneously arise in genomes of organisms all the time. You would expect a sequence for one of these self-cleaving things to show up every few million base pairs in a genome at random. And so there's no evidence that you need to inherit these things, that they can be developed de novo and have done so. The last possibility, and it's becoming increasingly clear that this is also true for some viruses, is that there are degenerate cellular parasites, meaning they used to be some kind of cellular parasite and have become so simplified that they are now considered viruses. And again, what's the step? I mean, you've got organisms like mycoplasma, like chlamydia, that go in and infect a cell using all the host stuff, right? All the small molecules, cofactors, all that kind of stuff. That last step requires you to open up your membrane and merge with the cytoplasm. That's really it. Then you don't need to make your own ribosomes. You don't need to make your own DNA polymerase or any of that kind of stuff because the host provides that. At that point, you've made the transition from a cellular organism to a virus. There have been a number of really, really big viruses discovered in recent times. Pox virus is pretty big, but here's Mimi virus. When this thing was first identified as an organism that infects amoebas, it was thought to be a gram-positive organism. because you can see it under a light microscope and it stains gram-positive. It looks like a gram-positive coccus. I mentioned it infects, it's bigger than a lot of known cellular organisms. The genome is 1.2 million base pairs. There are free living organisms with genomes smaller than that. And there are lots of parasites with genomes smaller than that. These things don't encode ribosomes, but they encode genes in the central information processing parts of metabolism. tRNA synthetases, the proteins that put amino acids onto tRNAs. Elongation factors, release factors, topoisomerases, translation initiation factors. These are proteins that are directly involved in the fundamentals of translation. There are even some that encode a few ribosomal proteins. I'll have that up here because it's more recently discovered. Of course, they don't encode any metabolism usually. They even keep that separation between host and parasite intact to some extent. There's not a membrane around it. It's not like it's still in a cell. But this is an example of Mimi virus. It's a crappy picture. But this is an amoeba, and this is something called the virus factory. There's a specific zone in the cell where viral genes are expressed, the proteins are synthesized, and the viruses are assembled. It's not membrane enveloped, but it's clearly this is virus and this is host. And so there is a distinction in the cell between self and non-self. That's pretty cool. All right, so this is a short one for today. That's all we're going to talk about. I'll remind you that there is no discussion session on Friday. I expect you to be on spring break, doing spring breaky kind of things. All right, great, thanks.